Hello, good afternoon, everyone. We are going to get started right away. And today's goal is to finish lecture 8 in the first half, and we're going to start on lecture 9 as well. Last week, we learned about how to solve various genetic problems, including monohybrids as well as dihybrid cross. Monohybrid cross is where we keep track of one gene at a time, and dihybrid cross is where we keep track of two genes at the same time. So let's start by doing a few more examples with the dihybrid cross. These are questions that are taken from the PowerPoint slides. Okay, so here we go. In bees, normal wings are dominant to vestigial wings. It actually doesn't really matter whether you understand what normal wings or vestigial wings means. A lot of people get a little bit confused when they find there are certain terms that they don't fully understand in the question, and then they don't know how to do the problem. But these are just traits, okay? They are different versions of wings. It doesn't actually matter what they look like, right? You just solve the question the same way. So for example, it says normal wings here are dominant over vestigial wings. So that's our first trait. And we can use capital N as oh, N's not working. Give me 1 second. Let me restart. We can use capital N for normal. Okay. Capital N for normal and small n for vestigial. Okay, if you're wondering what vestigial means, it just means the wings are really tiny and they're kind of shrunk a little bit. And the other character that we're looking at is the kind of pollen sacs these bees have. And it says in the question, deep is dominant over shallow. So we can use capital D for the deep pollen sacs and then lowercase d for the shallow ones. Okay. So that's the first thing you need to do in these questions. And you absolutely need to have pencil and paper with you as you're doing the quiz. You can't just like try to figure it out on the screen by looking at it. Then you're gonna have to see what the parents are. So of course there are two kind of parents, two parents here. We have the male bee, which has the vestigial wings and the shallow pollen sac. So if we write out the genotype, it is going to be small n. Because it is a recessive trait, it has to be pure. There are no other ways. That's why it wasn't necessary for the question to specify whether it's pure or hybrid. Similarly, shallow pollen sac is also a recessive trait, so we're going to use small d for that part. Okay, that's the male bee. Next, we are going to find the genotype for the female bee, which in this case is homozygous, normal wings, and heterozygous, deep pollen sac. So for the female, we would have big N. Homozygous means the same. Heterozygous would be different. So one capital D, one lowercase d, and that would be the female. Anybody have any questions about how to come up with the genotypes? Okay, so the next thing you have to do is to figure out the possible gametes for each of the parent. And here the male only has small n and small d, so all the gametes will be small n combined with a small d. On the other hand, for the female, it could be big N, big D, or big N, small d. If you forgot how to figure out the gametes, you can just take the N and try to pair up with the D. So big N, big N, small d. And if you repeat with the other N, you would end up having the exact same result. So we don't have to write it twice. Okay, moving on. Now we have the male and the female gametes. Our Punnett square will be 1 by 2, 1 row and 2 columns. We put the male gametes on the side and the female gametes at the top. Once you've done that, then you're gonna combine the alleles. So big N, small n, big D, small d. And you're gonna do the same thing for the other one. Big N, small d, small d. Right? So once you've completed the Punnett square, you have to answer the question, which is asking us to determine the genotype and the phenotype of all possible offspring along with the expected percentages. So big N, big D, small d, that's 50%. Right, that's this one right here. And in terms of the phenotype, they will have the normal wings and deep pollen sacs. Okay. The other 50% will be big N, small d, which would also be normal wings, but is pure small d, which means it will have the shallow pollen sacs. Okay, so that's how you do the question. All right, let's take a look at the next one. Just stop me if you want to ask anything, okay? Next question also has two characters that we are tracking. It's a little bit easier because the alphabets are chosen for you. So we have curly hair, which would be big C, and that's dominant over small c, straight hair. Right? It's a good idea to write it down even though it's already in the question. And then it says albinism is a condition in which cells which normally produces pigment does not do so. A person with this condition is called albino. The allele for skin albinism is recessive to normal allele. So big N for normal, small n for albinism, okay, or albino. So that's the alleles and what they represent. Then we're gonna take a look at the family here. We have the mother. She has curly hair and albinism. And for the dad, he has straight hair and normal skin. Okay, and then we have a kid as they have a kid who has straight hair and is an albino. Okay, so to organize the information, we can draw a pedigree, simple one. That's the mother, that's the dad, and then they didn't tell us whether the kid is a boy or girl, so we just put like diamond. So let's start with a woman. She has curly hair. Curly is dominant, which means one of the two alleles would be capital C. But at this moment, I don't know if she is big C, or big C, small c. So I'm just going to leave it as a blank. She has albinism, which means it is going to be small n, small n. The only way to express a recessive phenotype is to have pure genotype for that particular trait. Now, for the for the men, he has straight hair. Straight hair is recessive, so he must be small c, small c. And then he has normal skin pigment. Once again, we don't know if he is big N, or big N, small n. It's one of them. Let's take a look at the kid. The child has straight hair, which is small c, and he is an albino, which means it's small n, small n. So by looking at their child, we can actually work backwards and figure out the missing allele. The child has two little c. One little c came from the dad, the other one must have came from mom, right? So this one of the little c must have came from mom. So the mother has to be heterozygous for the, hair gene. Similarly, for the one of the little n, one came from mom, the other must have came from dad. Okay, so that's how we figure out the missing genotype. Generally speaking, if a person has a dominant phenotype, you cannot tell immediately if they are homozygous or heterozygous. You would have to look at either their kids or their parents, or sometimes looking at their siblings to help you figure out, um, whether it's pure or hybrid. Any questions before we move on to more stuff? All right, thank you, Michael. Anyone else? All right, I am just gonna copy more questions over. How many unique gametes could be produced through independent assortment with this many genes? So there are 5 genes here. How many gametes can we produce? Well, we did this before, right? Last week. All you have to do, you don't actually have to list all of them. You just have to take a look at each gene. So for example, the first gene, there is a big A, there is a small a, so 2 possibilities. Big B, small b, 2 possibilities. Big C, that's actually just one option, right? You can only pass on the capital C. As for the D, 2 options, and for the E, 1 option. So if you want to see the total number of combinations that can be created with this, you multiply all of them together, and that will give you 8. 8 different gametes. Okay? How about this one? How many combinations can we create? 32 is correct, is 2 times 2 times 2 times 2 times 2. This time everything is hybrid, so there are 2 options for each gene. And if you multiply all of it together, you would get 32. Next question. Consider the cross this and that. What is the probability of producing a genotype of that? Okay, so if you try to come up with all the gametes, the table is too big. It's going to be like 8 by 8, okay, which has 64 different outcomes, and that would just be like way too much work for multiple choice questions. So instead, what you do is you just look at each gene individually. So if you do big A, small a, big A, small a like this, Okay. And you do the Punnett square. I mean, you should have probably have this memorized by this point. And we're looking for big A, big A. That's what the question asks. So what percentage, what's the probability of getting big A in this case? Well, it's 1 out of 4, right? Now, similarly, you can switch the letters A to B, which would also give you 1/4. And then you can do it for C as well, which is another 1/4. And if you want to figure out the combined probability, you would multiply them together. Just like if you want to figure out the combined combinations, we multiply. Okay. For what we are doing, if you're confused whether to add or multiply, most of the time it's multiply. Okay. If you absolutely have to guess which one is it, it is just multiply. So quarter times quarter is 1/16 times another quarter, that's 1/64. Okay. Now, just on the side, what if I ask you for A, big A, big B, small c, small c? Well, think of the Punnett square. Big A is 1/4. What about big B, small b? What's the probability of big B, small b in the Punnett square? Again, with the same parents, okay? Big B, small b. Big B, small b would be like this one, right? Which is 1/2. Small c is like this one, which is 1/4. So if you multiply these together, it is going to be 1/32. Okay? If you reduce 2/64, you get 1/32. So that's how you do it. Yeah, 2/4 is fine. 2/4 is same as 1/2, right? Mhm. Yeah. All right. Here's another question. Let's take a closer look. True breeding. True breeding basically means pure. It's another way of referring to it. So two true breeding stocks of pea plants are crossed. One parent has red axial flower and the other has white terminal flower. So basically we have red versus white. We don't know which one is dominant yet. And then the other one is axial flower versus terminal. Again, you don't have to understand what these actually means to finish the question. Okay, there are just two kinds of flower. One type is called axial, the other is called terminal. Okay, if you're interested, terminal means the flower is at the end. Okay, axial means the flower is like coming out on the side. Okay, that's all it means. Okay? But like I said, it doesn't really affect how you do the question. All right, so we don't know which one is dominant, which one is recessive, which is why we have to look at the F1s. Right? Remember the mouse thing I did with you guys last time? If the parents are pure, all you have to do is look at the F1s to figure out which trait is dominant. So all the F1s are red, which means red must be dominant. Okay, this is the dominant one. And all of the F1 have axial flowers, which means that's also the dominant. Another thing to remember is that if the parents are pure, then all F1 will be hybrid. So F1 will be big R, small r, and big A, small a. Red, and with the white allele that's not showing up. Axial allele with the terminal allele that's also not showing up. If 1,000 F2 offsprings result from cross of two F1s, so we're taking an F1 crossing another F1, but the F1s are hybrid for both genes, then that fits our criteria of the 4 by 4 Punnett square. Let me just quickly show you what we did last week. Right, we did this 4 by 4, right? And the takeaway from this 4 by 4 table is that the ratio will always be 9:3:3:1. It will save you time on the test to not have to create that table. And what is the question asking? Of the 1,000 individual, how many do you expect to have red and terminal flowers? Okay, so red is dominant, and axial is dominant. That's this one, DD, right? Oh no, I am so sorry. I read it wrong. Okay, let's go one more time. You, uh, you're asking for red, which is dominant, and terminal. Right? Terminal is recessive. So 9 is dominant, dominant. These are one dominant plus one recessive. Okay? Doesn't matter which order. And then the one at the end is recessive and recessive. So if we're looking for dominant combined with recessive, the ratio would be the number 3. And if you express that as a fraction, it's 3/16 times 1,000. And that's going to be what, 180, I don't remember, 82.5 or something like that. Let me just plug that in here. So uh, in case you don't know how to do it, 3 times 1,000 divided by 16. Okay, so that's 187.5, right? But because it's not like exact, right? Like I said, these are just probabilities. That's why you wouldn't find 187.5 of a flower. So you just find the closest number, which is what Uthman said before. It's 190. You guys understand this? Okay, so if you see a question like this on the quiz, just use the 9:3:1 ratio. As long as the parents are hybrid for both genes. If it's not hybrid for both genes, like the B thing that we did, right? The parents are not hybrid for both genes, and you can't do the 9:3:1 ratio. You have to physically create a table. But that's fine because this table is small, right? It's only 1 by 2. Okay, you should be able to create that. The quiz, I give you guys more time this time because there's a lot of problem solving you have to do. Speaking of which, the quiz is supposed to be next Tuesday originally, but I am not confident I can finish the lesson by Thursday. I probably will, but just in case, I'm gonna postpone the quiz from the Tuesday to the Thursday. Okay? So we do as much as we can on Thursday for lecture 9. We'll wrap up whatever we can on Wednesday. And then next week is actually the last week before the final test. Okay, so we just have 2 more lecture to go, number 9 and number 10. 10 is really short. 10 can be finished within one class for sure. Okay, that's that. Any last minute thing for dihybrid before I move on to new things? Great. So everything we learned so far is, let's call them like regular, dominant, recessive, inheritance. Okay. And what that means is like if you have big R for flower, then this would be a red flower. Small r would be white flower. And big R, small r would be red because the big R is going to cover up the small r, and the flower is going to show up to be red. So nothing that you don't already know. There are two other kinds of inheritance pattern that you need to know. First one is called incomplete. Incomplete. Incomplete dominance. Incomplete dominance. So in incomplete dominance, big R is still red. Small r is still white. But the heterozygous condition is not red and is not white. Incomplete dominance means the allele does not completely dominate over the other one. It does not cover it up like the regular case. In the regular case, the big R completely dominated over the small r. You won't even see the small r showing up. It's going to be red. But in this case, big R doesn't cover up small r, and small r does not cover up big R. And in the end, the heterozygous condition is going to be somewhere in between. So somewhere between red and white is going to be pink. Okay. We also have another inheritance pattern called codominance. By the way, like for a particular character, it's either this, or this. It cannot be all at the same time, okay? Like it has to be a particular mode. So for codominance, big R is red, small r is white, okay? And big R, small r, in this case, rather than creating a blend in between, codominance means both coexist. They both get expressed. So this would be like a white flower with some red dots or a red flower with some white dots. Okay. And there is a slide for that in, oops, there's a slide for that in the PowerPoint slide. Okay. You can see codominance, both showed up, and incomplete dominance, it's a blend in between. So human blood type is an example of codominance. If you inherit type A from mom and type B from dad, you're going to be type AB. Okay? We'll learn more about blood type next semester in the second week, actually. All right. Now, my other classes were a little bit confused with these things. So let me see if you guys can answer the question. Yeah, that's possible, right? But may not be. Okay, maybe they have a separate gene for each section of the wings. That's actually quite possible as well. I don't actually quite know. All right, so here we have a mouse with let's do a short tail. And then we have another mouse with long tail. Okay, and then we cross them. They are both pure, okay? Pure. And then we cross them, and then the babies, okay, they have medium tail, medium length tail. Let's do a poll here. We don't have a lot of people, but let's do a poll anyways. What is this an example of? Is this an example of incomplete dominance or codominance? There are only two choices. Short tail, cross with long tail, and then everybody in the F1 has medium tail. Is that A, incomplete dominance, or B, codominance? Okay, so similar result as my other class. It's kind of 50-50. This is actually um, incomplete dominance because you get something in between. It's not long, it's not short, it's somewhere in between. If it's somewhere in between, it's incomplete dominance. A codominance version of this would be, the mouse have like two tails, a long one and then a short one. But that's not what happened, right? Okay. So remember, codominance means both will show up. Incomplete means somewhere in between. Understand? Okay, let's try to solve some codominance and incomplete dominance questions. So the question will tell you what kind of inheritance pattern you're dealing with. For example, here it says in snapdragons, which is a kind of flower, red is incompletely dominant over white. Right? And then it tells you the heterozygous condition produces pink flowers. What will be the result of a cross between two pink flower snapdragons? Okay, so we know already heterozygous is going to be the pink, and they're asking us to cross two pink flowers. So we have big R, small r, cross big R, small r. These are both pink, pink and pink. And the way we do the Punnett square is the exact same way from before. What is different here is how you interpret the results. Right? So we get our Punnett square done like that. And in this case, we will have 25% red and we will have 50% pink, right? And finally, we will have, what is it? 25% white. There you go. 25% white. All right, that's all. So how do I guarantee I produce pink flower? Right? It's kind of counterintuitive for at first glance. Like you take a pink flower and you cross a pink flower, and actually only half of them would be pink. So what parents should we start with to guarantee the highest yield of pink flower? Well, interestingly, yeah, that's right, Michael. If you take a pure red and you cross it with a pure white, that will create 100% big R, small r, or pink. Okay. And that's how incomplete dominance work. All right, let's do another example. A red bull is a male cow, crossed with a white cow, the female, produces a roan calf. Calf would be a baby cow. A roan calf has intermingled white and red hair. So this would be an example of codominance. It's not, red hair or white hair or somewhere in between. It is intermingle. It's both, right? Both red and white, and you can see there are patches of white and red on the cow. And the question is asking us, if a roan bull is crossed with a white cow, what percent of the calves would be roan? So roan bull is big R, small r. That's the heterozygous condition. Cross with a white cow, which is little r, little r. And again, you can go ahead and do the Punnett square. Same way as before. And it will be 50% roan and 50% small r, which is white. And so I think these are much easier than the dihybrid cross questions. But on the test, they are all mixed up, right? So you have to be careful and decide which type of questions you're dealing with. All right. Everything we've done so far, whether is dominance, recessive, incomplete dominance, or codominance, they are autosomal conditions. Okay, they are transmitted on the autosomes, which are chromosome 1 to 22 in humans. Some conditions are transmitted on the X or the Y chromosomes, and we call these sex-linked conditions. If it's on the X, then both males and females can get it. If it's on the Y, then only males can get it. Now, there are different kinds of X-linked disorders. Some are dominant, some are recessive. If it's X-linked dominant, then more females will be affected than males because females have two copies of the X. So if they inherit one copy of the X, that's enough to create a condition for them. However, if it's a recessive X-linked disorder, such as hemophilia, which is a bleeding disorder, your blood don't really clot easily, or red-green color blindness, then more males will get it compared to the females. Because female has two X's, and if this is a recessive condition, the normal X is going to protect the female from a disease X. Okay, so these are recessive condition. Okay, both of them are. So let me write down in case you missed it. X-linked dominant, female more likely to get it than males. If it's X-linked recessive, then males are more likely to get it than females. All right, here is a test for color blindness you probably have done before in like a eye doctor's office or something. It's called the Ishihara test. What number do you see? Do you see 74 or do you see 21? Yeah, if you see 74, that means you do not have color blindness. If you see 21 and you did not know you have color blindness, you might have color blindness. You probably want to check that out. Okay, so this is a very quick and easy and non-invasive way to determine if someone has red-green color blindness. All right, next we're gonna try to solve problems involving X-linked conditions. I'm just going to use color blindness as an example. It's easier for people to understand, it seems, compared to something that they might not have heard of, like, hemophilia or something like that. Okay, but you solve the question the same way. So let's consider color blindness, which again is an X-linked recessive condition. It's X-linked, meaning it's transmitted on chromosome X, and it's recessive, meaning you need to have two copies of the bad X in order to have the condition. If you're female, if you're male, then you get the disease X, then you have the condition right away because males only have one X. So let's draw these out, and we have a square here, and that would be a normal male. Okay, when I say normal male, I mean a male that has normal vision, okay, in the context of what we're talking about. So his genotype would be XY. Okay, let's draw an other male, but this time we're going to shade it in. So this would be a colorblind male. The genotype for a colorblind male would be X with the superscript of C to indicate this is a colorblind gene on the X and Y. Because male only have one X, if they inherit the colorblind X, they are going to be colorblind. This would be a normal female, normal vision female. And be X and X, two normal copies of the X chromosome, no disease. We're going to go ahead and shade in the circle. This would be a colorblind female. And both copies of the X chromosome will have the colorblind gene on it. And we have a last case here where we only shade in half the circle. This is a normal vision female. She has one copy of the normal X and one copy of the colorblind X. But because colorblindness is recessive, the normal X will override the colorblind X. Okay, so briefly, colorblindness is basically failure to produce a particular protein in the eye that allows you to detect certain colors. So to make that protein, you need the gene on the X. So if one copy is not making the protein, that's okay. You have a backup copy that will make it. It's only when both copies are not available to make the protein, then you can't see those colors. So we call these carriers. Okay, so a carrier is a female that has the gene for the disease, but does not actually have the disease herself. Can there be a mutation in this? Yeah, this is caused by a mutation, okay? But it's not something that you will get in your lifetime. Okay, like if you're not born with it, then you're not gonna catch it, or you're not gonna mutate and all of a sudden have color blindness, right? But it is an inherited mutation from the previous generation.
Oh no, so like what I mean like is like how we learned the incomplete and the codominance. I was wondering if there's like a mutation where any of those could possibly occur in that.
Uh, Sorry, one more time. I don't quite understand. One more
Yeah,
time.
all good. So you know how we did the incomplete dominance and how that equals pink and the codominance where equals white plus
Yeah,
red?
yeah.
Is it possible where that could equal like one of those instead of just a regular dominant recessive inheritance?
Oh, for color blindness?
Yeah.
No, for color blindness it's just dominant and
Oh, okay.
recessive straightly. Yeah. Okay, can a male be a carrier then? Right? Can a male be a carrier? That's right. Male cannot be a carrier because males only have one X. If you have the disease X, you're gonna have color blindness. Right? Carrier means we don't know you have that gene. Um, but because you're not showing the disease, okay, but you're carrying it. All right, so those are all the possibilities. Let's do some of the questions in example 8. The first one is carrier female cross normal male. Just make this a little bit smaller. Carrier female cross normal male. Okay, so carrier female is XXC and normal male is XY. And we would just do the Punnett square the same way. Okay, XXC, we're going to separate the chromosomes. That. And we will also separate the chromosome here for the father. Half the sperm will get X, the other half will get Y. We will put these together. Similarly, we'll put these together. Now, when we report the genotypes and phenotypes for sex-linked stuff, we generally do it separately for the females and the males. Right, so these are the daughters or potential daughters, if they're going to have them. And then these are potential sons that they might produce. So in terms of the daughters, all of them will be normal vision. Okay, all of them will be normal, even though if like there's a 50% chance that she will be a carrier, but doesn't matter. She's gonna have normal vision. For the sons, though, okay, remember we're only looking at the sons, just the blue stuff. 1 out of 2 sons, or 50%, will be okay, will be normal. But 50% chance for the sons to have color blindness. Okay, you see, this is an X-linked recessive trait. Males are more affected than females. Any questions? If we're not being gender specific, then you can say there's only 25% chance that any of the any of the children will have color blindness. Okay? But if we're being gender specific, only the boys and only half the boys will be affected. Okay, moving on. You tried this one and tell me what you get.
Sir, can you tell me again why males are affected than well,
Yeah,
female?
only if it's recessive though, okay? Only if it's recessive. Males are more affected than females because female has an extra X chromosome. So look, this person inherited the disease, the colorblind X, but she has a normal X that overrides it, which means for female to have color blindness, she needs to have 2 copies of the colorblind X. Right? So in a way,
Mm-hmm.
it's more unlikely for them to get it, right?
Mm-hmm.
But you got to understand this is switched if we are talking about a dominant condition. Because if it's a dominant condition, then the extra X actually becomes a liability because now you have a greater chance of inheriting a bad X, right?
Okay,
Yeah,
thank you.
no problem. It's kind of like this, right? Like let's say there's a bag, okay? It's a bag of balls, okay? And there's a red one in there. Okay, there's a red one in there. You cannot see the balls, okay? If we are talking about a dominant trait, because male only have one X, then we would stick our hand in and we would take out one ball, and that's it. Okay? So if that ball is red, then we have the disease. If it's white, we don't have the disease. But for females, because they have two X's, they will have to take out two balls, right? And so you increases the risk of catching that for dominant trait. Okay? And for recessive, it's okay because the normal X is going to cover it up. It's kind of like that. Okay, please work this out and let me know. Let me know what you get. Right. Let's put the father here on the side. Again, it doesn't matter. We can put him on top or put the mom on the side. It doesn't matter. So what do we have? Well, for the daughters, they are all going to be okay. And this again shows you why if it's recessive, it's harder for it's not harder, but like unlikely for the females to be affected. Okay, so daughters, 100% normal. They will all be carriers, but they will not have color blindness. For the sons, everybody will be colorblind. Okay, that's all. I mean, there are two more examples which you can, solve on your own. Um, but I think two examples are good enough for now. All right, here's another question. The pedigree below shows family members who exhibit a dominant trait. What is the probability that the children of 4-3 and 4-5 will have the dominant trait? Okay, so it already tells you in the question that this person is big W, small w, and this is small w, small w. And so if we look at individual 4-4, she does not have the trait. That means she must be small w, small w. For individual 4-3, he has the trait. So he will have capital W and something else. The other thing could be a big W, could be a small w. We don't really know. That's why we have to look at other people in his family. Okay? So if we trace back to the mother does not have the trait. We're not sure what the trait we're talking about here is, but she doesn't have it, so she's going to be small w, small w. Okay. This is not sex linked, right? By the way, it's just regular on the autosomes. So if the mother is small w, then this person will definitely inherit a small w from his mother. And so we know 4-3 is going to be hybrid and 4-4 is going to be pure and not have the trait. And we want to look at their kids, which means we will set up the Punnett square. And the question wants to know what's the probability that the child will have the dominant trait? And that is going to be 50%. Okay. So that's half of the quiz for next Thursday. The other half will be on lecture 9, which we will start in 5 minutes. Okay, so let's take a quick break. And when we return, we will do lecture 9. See you guys in a bit. All right, so for the second half, we are going to be talking about biotechnology, right, which is a little bit harder than lecture 8 because of a few of the concepts are a little bit abstract. But I'll try my best to explain it to you. First of all, what is biotechnology? Biotechnology is technology based on biology. Right? Basically, you learn about how a cell works. Right? That's what we've been doing since the test number 1. We learned about, how DNA are replicated. We learn about how transcription translation occurs. And what scientists wants to do is once they understood how these processes occur within a cell, they try to control it. They try to alter it and harness the cellular and biomolecular processes to create some kind of technologies that will help basically improve the lives of humans. And there are many different kinds of biotechnologies. We're not going to have time to, go into details for all of them. I will pick and choose a few to explain to you. The most common technologies that are being used and that has been used for quite some time is what we call recombinant DNA. Recombinant DNA is basically a chunk of DNA that is made up of DNA from different sources. So if you take DNA from a jellyfish and then you put it into the DNA of a mouse, the end result would be a recombinant DNA. And that's what they did here with this in this picture. Right? The mice you see are not, these are not Photoshop pictures. These are real pictures. The mouse, sorry, the mice. English is not my first language, and sometimes these plurals get me all confused. Anyways, the mice in the picture, they are able to glow in the dark because they have the jellyfish gene inserted into them. These jellyfish, of course, are able to glow naturally, and that's because of a protein called the green fluorescent protein. Right? So it's not like they inject the gene into like adult mice and then all of a sudden they glow. This has to happen in the embryonic process, right? Before the fetus is formed, this has to occur already, and then all the cells inside the body of the mouse will have the GFP protein, and then it's gonna glow. Right. So the scientists do this not just for amusement purposes. There are real application of these kind of stuff. GFP is actually very useful in a lot of research settings, and if you're interested, you can look it up on your own. We don't have time to go into all of it. In terms of recombinant DNA, I will go through two specific examples with you where it is applied in medicine as well as agriculture. Starting with agriculture. Right? Golden rice. Has anybody heard of golden rice before? Again, we have a few people here. Anyone seen golden rice before? No? All right. So golden rice is a genetically modified organism or GMO, right? So I'm not gonna tell you whether GMO are good or bad. You can have your own opinions. I'm just gonna tell you what it is and what it does in this particular case. So vitamin A deficiency is a common condition in low-income countries. It is the leading cause of preventable childhood blindness. It turns out vitamin A is needed to make certain pigments in the eyes that will allow you to see. And if you are severely deficient in vitamin A, it could lead to blindness quite early on in life. Lack of vitamin A also lowers immunity, and in some of these low-income countries, they often don't have very good vaccination rates as well. So a poor immunity with poor vaccination rate can actually encourage the spark of an endemic for infectious disease. We are all familiar with a pandemic. Pandemic is like global. So an endemic is local. Right? And one level up from the endemic, where it's more regional, that's called epidemic. Okay? And then when it's all over the world, pandemic. Right? So then in some countries each year, it is estimated that vitamin A deficiency is related to 670,000 deaths in children under the age of 5 and cause an additional 500,000 cases of irreversible childhood blindness. So obviously, this is a serious problem and it needs to be addressed. And it's not as simple as, why can't we just feed them more vitamin A? It's not very easy to provide food that are rich in vitamin A to them because it might not be part of the diet or the food might be very expensive once it gets to the local markets because it's not grown natively in their countries and you have to fly them in and access and all that stuff, right? So there's no easy fix, and that's why people created the golden rice. What is golden rice? Oh, sorry. Before that, this is just a heat map that shows you where vitamin A deficiency is quite prevalent. So it's not a problem in high-income countries, but it's uh, pretty much a very severe clinical condition in low-income countries, as I've mentioned. So golden rice is genetically engineered rice that is created to make beta carotene, which is a building block for vitamin A. You must have heard your parents said this to you at some point in your life. Eat more something, it's good for your eyes. What am I talking about? Yeah, carrots, right? Okay, eat more carrots. It's good for your eyes. It's not completely untrue, right? Because carrots contains beta carotene, and it is used as a building block for vitamin A, which again is necessary to make certain pigments in your eyes to help you see, particularly in the dark. Okay, so rice by itself does not have beta carotene. They are often white or brown, but in this golden rice, um, because it has been genetically modified, it produces quite a bit of beta carotene. Okay, and rice is actually a staple food in a lot of these low-income countries, and so it's um, a good choice of food to put the beta carotene in. Right? You can store rice for a very long time. They don't really spoil if it's kept dry, right? You can transport them in bulk, right? So it's a logical choice for something like this. So it's a recombinant DNA because the rice DNA has been altered and there are two additional sources of DNA that has been incorporated into the rice DNA. One is from daffodil. The gene they took, it's called site. We usually use italics to represent the gene. And there's another gene that has been extracted from soil bacteria. And you put these two things into the rice DNA, and they have the means to create beta carotene. Okay. People who support the use of beta carotene, support the use of golden rice, argues that it addressed the vitamin A deficiency crisis in some of these low-income countries. If you can prevent a child from going blind, why wouldn't you, right? It will also improve public health because, as I told you, vitamin A deficiency is linked to low immunity. So if they have the golden rice, it will improve public health in that regard. And it also enhance food security because not only are you supplying the vitamin A, you're also supplying them with food, the rice itself. So these are arguments for the side that supports the use of golden rice. It's, by the way, it's already in circulation. Okay, it's not experimental or anything like that. It's already out there. People who opposes the use of golden rice argue that there could be unintended consequences. Okay, not just in the people who are consuming the rice, but also, in places where you grow them. Right, what happens to the rice if it gets out into the wild, right? Is it going to become an invasive species? Is it going to affect other crops, right? So they believe there are not enough research being done to assess these kind of environmental impacts as well as potential long-term negative impacts in people who consume them. They're also worried about corporate control and patterns, right? Obviously, these are very valuable intellectual properties created by various companies, and you never really want to have these powerful companies being the sole source of a particular food type in low income countries, right? There are potential financial and as well as political uh, uh, impacts, right? From these kind of corporate presence in low-income countries. Also, socio-economical factors, right? That has to do with access. Once you get the golden rice to these countries, how do you distribute them equitably, right? It's all legitimate concerns for people who are opposing the use of golden rice, but I personally don't think is a unique problem to golden rice. Okay, it's a common problem for many things, and like many other problems, it needs to be addressed. But once it's being addressed, I think the benefits outweighs the drawbacks here. Okay, so that's recombinant DNA applied in agriculture. Any questions? Wonderful. Next, we're going to talk about the other application, which is in medicine. Right? There are so many medical application of recombinant DNA. We only have time to talk about one, and I chose an easy one for people to relate to. Okay? It is the case of insulin. So insulin is a hormone, and it was discovered by Frederick Banting and Charles Best. These are Canadian scientists. Actually, they have a building dedicated to them uh, at downtown UT campus. So, what does insulin do? Insulin basically helps you absorb sugar from the blood after a meal. If you don't produce insulin or if you don't respond to insulin, then you eat and the sugar stays in the blood and it doesn't go into the cell. So your body thinks you're starving. And as a result, it starts burning your body fats and body protein, and you basically waste away your body mass. If you burn a lot of fat in a short period of time, you can get ketosis, which cause your blood pH to drop, and that could be quite dangerous. Right? So people um, if they have unhealthy lifestyle, okay, later on in life they might get type 2 diabetes. But this is not what we are focusing on. We are focusing on type 1 diabetes. People with type 2 diabetes, you cannot give them insulin. It's not going to do anything. Okay. Exactly why, we'll learn that next semester. But for people who have type 1 diabetes, these are diabetes that occur early on, usually in life. It can happen sometimes later on as well, but generally it happens earlier in life, and people might even be born with type 1 diabetes, and their body is not able to make the insulin. Okay, so as a result, they need to inject insulin into their body to help regulate the blood sugar. So before biotechnology was available, the only way to create commercially available insulin were to harness them from cows and pigs. Okay, they take the pancreas from the cows and pigs, grind them up, and then they try to extract the insulin from it. Right? And why do you think that might not be ideal? Like if you are someone who needs insulin, what are some of your concerns if I tell you it's taken from cows and pigs? Anybody? No concerns. Just gonna be happy and take it. Anyone at all? Anything you can think of? Probably what happens to animals, or even just that is from animals. Yeah, okay, so sure, right? Some people might worry about uh animal handling, right? Some people advocate for animal rights and so on and so forth. That certainly is a legitimate concern, right? Yeah, getting diseases from it, right? Purifying the compound might be complicated, and there might be impurities in it, right? And perhaps the most concerning thing that you might have is that these are from the animals, and the animal version of the insulin is not necessarily 100% same as the human version, and there could be some efficacy problems, right? And if it's taken from pigs, then because of religious reasons, some people might not be able to take it, right? So, there's lots of problems basically, and not to mention it's expensive and time intensive to prepare these things on a large scale, which is why we have the biotechnology, right, method of creating insulin, and that was done in the late 20th centuries, and that's what I'm gonna show you. In order to do that, in order to basically turn bacteria into insulin factories, there are four steps we need to do. We have to locate the gene of interest. We have to find out where the gene is, okay, in the human genome. Once we found the gene, we have to clone the gene. Okay, we have to clone the DNA. We have to make a lot of copies of just that gene. Then we're going to feed it to the vector. In this case, we're going to feed it to the bacteria. So the bacteria will have the insulin gene, and now it is a recombinant bacteria because its genome not only does it have the bacteria genes in it, there's also human genes in it. Is a recombinant DNA. And then nature will do the rest. The bacteria will pump out a lot of insulin. This is a plate of bacteria, and the blue ones are the ones that has taken up the DNA. Okay, those are the ones we want. Okay, we can literally, if you can see it on a plate like this plate is like no bigger than the tissue roll here. Okay, if you can see the little white spot and blue spot with your naked eyes, that means there are literally millions and millions of bacteria in that tiny tiny circle. So if you scoop up one of these things and you put it in a new solution, they will start growing and growing and growing, and they will produce a lot of insulin for you. Right? So that's one way of telling whether or not the bacteria has accepted the DNA. That's not the only way, as we will see later on. Okay. I think today we will only have time for number one and number two, and then on Thursday we'll finish with three and four. So here we go. Locating the gene of interest. Where in the human genome, right? Where in the 23 pairs of chromosomes will you find the gene that you need? Right? This used to be an impossible task. It would take, decades of research sometimes to figure out the gene of interest. But thanks to what's called the Human Genome Project, now anybody with an internet connection can actually look up any gene they want. I'll show you that in a second. And this Human Genome Project is basically an international collaborative effort to map out the entire genome of humans. Okay, they wanted to know all the ACTGs, all 3 billion ACTG base pair on every single chromosome in the human body. It was a very ambitious project. It took over a decade to complete. Okay? It started in 1990, and then it's done in completed in 2003, right? So that is over 13 years to finish. Okay, I remember when I was in high school, the Human Genome Project was like my report. We had to do a report for biology, right? So at the time they just started, and now it's all completed and it's freely available online. So there's a website called Human Genome Browser, which you can play around with if you want. But it's not just humans. They actually done it for like so many organisms now. Okay, and they're getting better and better at it. Okay? They are able to do it very quickly nowadays, and with fewer resources. Right? If you want to sequence the genome of an organism, it's definitely not going to take a decade now. So like, they have cats, they have a bunch of things that I don't even know what they are, and chickens and whatnot. But anyways, we are looking for humans. I lost the human thing here. Where's the human? Okay, there were humans. And we can look for any genes. We learn a lot of genes. I mean, we didn't learn. We learn about what a gene is in the course, but we actually know some names of some of the genes, right? I was gonna ask if anybody remember any, but I think I'm just gonna tell you. Okay. For example, we have this gene. Do you remember this one? BRCA1 gene. What kind of gene is this? Can someone tell me? Give me some encouragement, guys. Okay, this is very challenging.
Breast cancer,
Thank you. This
broccoli.
is the breast cancer gene, right? And it's a tumor suppressor gene that helps repair DNA. All right, so let's search them up. Da, 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 da. And there we go, BRCA1 gene. It is on chromosome number 21, and it is base pair number 4,304. No, I read that wrong. 44,295, all the way to this. That's how long the gene is. And you can click on it, and it will show you the exon and all the stuff in between. Those are the introns, and then there are different variations of the BRCA1 gene in the human population. Okay, and you can see it is on chromosome 17. This is the centromere, and this is the shorter part before the centromere is called a P arm, and then the longer one is called a Q arm. And right here, that's where you will find the BRCA1 gene. It's the same location in you as it is in me. Okay, males also have the BRCA1 gene. Okay, uh, because again, like it's responsible for DNA repairing, right? All right, I'm gonna click into the BRCA1 gene, and you can click on the sequence, genomic sequence. There's a bunch of stuff that, we don't have to worry about. I'm just gonna press submit. And there you go. This is the BRCA1 gene. Okay, all the ACTGs, right? GCT, GAG, ACT, and you can actually transcribe this and translate it, and you can get the protein from it. And you can see we have capital letters, and then some of them are lowercase letters. The capital letters are actually the exon. So if you do the transcription, the lowercase will get cut out, and those are the introns, right? And there is your whole G. Right? A lot of it is just filler. You can see a lot of it is lowercase, right, which will get cut out, right? So that's the BRCA1 gene. And we know this for all the genes in the body. Okay? They have it all on the internet, right? Which is kind of cool, I think. What else can you look at here besides you can also look at the protein structure, maybe protein structure. Do they have protein sequence here? Sometimes they do. Um, just click on that, and then it will take you to another place. And if you wonder, oh, they don't have a picture. Sometimes they do. Okay. Anyways, so that's how you find a gene, right? So now I know it's on chromosome 17. Then the next step is to clone it. Right? Look, you have to locate a gene, and then you have to clone it. Clone it means make many copy of it. Right? So all you have to do is actually supply a few human cells, and then you don't want to copy all the DNA. That's too much. Okay, you only want to copy what you want, which would be on the chromosome 17. And the technique to do it, the biotechnology that's needed to clone DNA, is called PCR, which stands for polymerase chain reaction. I'm sure you've heard of PCR before, uh, just especially during the pandemic, right? Because that's the test that you would need to get earlier on to see if you're positive or negative, right? Yeah, eventually they came up with those little kit that you can take home and test yourself. But at the beginning, it's PCR test. And nowadays, you might still need to do a PCR test to be 100% sure in some cases, right? So we'll talk about that in a second. But the PCR has been around for a long time, even before COVID, right? And it allows us to copy a specific sequence of DNA as many times as we need to. So there's a machine that scientists can buy and it's really small. It's like a coffee maker, okay? And you would put your DNA sample in a little um, tray with whales in it. You would add your reagents, add your DNA, and then you put it in a machine and set it up, and then it will copy exactly what you want it to copy, right? And it runs in cycles, and every cycle is going to double the amount of DNA. So let's say you start with 2 copies. If you run it one time, then you have 4 copies, and then you would have, 8 copies, and so on and so forth. If you do it 30 times, this is a really big number. Let's see, 2 to the exponent 30, right? You're gonna have billions of copies after 30 cycles. This doesn't take too long, right? It usually takes about 2 hours, right? When I used to do PCR for research, I would set up in the morning and then I go do other stuff and come back 2 hours later. And you have billions of copies of the DNA that you want. So how does it actually work? The method is based on the understanding of DNA replication. We learned that, right? We understand the process of DNA replication involves using gyrase and helicase to separate the DNA strand. That's the first step to create a replication bubble, right? But in PCR, instead of using gyrase and helicase, we use heat to separate the DNA strands, right? Depending on the composition of the DNA nucleotides, if you're more C's and G's, you need to heat it up more. If you have less C and G's, then maybe not so much. But typically around 95 degrees, plus or minus, depending on the CG content, you will be able to separate the DNA strands. Okay? Once you separate the DNA strands, in your solution, you will have to create primers. Okay? Primers are basically same as the RNA primer, but we're using DNA primer here. And it allows you to decide what part of the genome you want to copy. And since you have the sequence, which I have lost, give me a second. Let me try to pull that out again for you because it's important for you to understand what I'm saying. Genomic sequence. This time, let me just take away the introns. Okay, submit. There. So this is the whole gene. It's much shorter now without the introns. And I would create a primer that would bind to the beginning, and I would create a primer that binds to the end. Then when we use the polymerase to copy the DNA, it will only copy between the primers and nothing beyond that, right? So it's like, I give you a textbook and I ask you to photocopy some pages for me, and then I put 2 sticky text in it, right? So copy everything between the sticky text. Make sense? The primers are the sticky text. Okay. So then if this is the gene, then I would create the primers that are complementary to the beginning and the ends of the gene. And normally in a cell, we would have DNA polymerase 3, which would be doing the actual copying. But because we have to heat things up to quite high temperature in PCR, we cannot use the DNA polymerase 3. Okay? It's going to denature the protein and nothing's going to get copied. So scientists discover some bacteria live in hot spring, right? And these hot springs are really hot. They go up to hundreds of degrees sometimes, right? You probably have seen pictures like this before. And those bacteria actually also has polymerase in them because they too need to copy their DNA. And the scientists have extracted the polymerase from them, and it's called tag polymerase. Okay, tag polymerase. They don't really extract them like anymore. Okay, they've extracted at the beginning and studied it, and now they can synthetically create it in the lab. Yeah, you can actually buy them as powders, and you can reconstitute them. Very convenient. And so that's what's going to be copying the DNA. We have the template here. Uh, as I show you, we have the primers, and then the tag polymerase will copy everything in between. Okay, moving on. The polymerase chain reaction actually has 3 steps. We heat it up. That's called denaturation, which is to separate DNA. Okay. around 95 degrees plus or minus. And then you want to lower the temperature a little bit in the annealing phase. By lowering the temperature a little bit, you allow primers to attach. Okay, you don't want to lower it too much. If you lower it too much, then all the DNA will just come back together. Right? We just want to lower it a little bit for the primers to bind to its target. Because the primers are complementary, it will seek out which portion to bind to and ignore everything else. Finally, we want to heat it up a little bit to activate tag polymerase. Okay, because it's a hot spring bacteria protein, it's if the temperature is too low, it's not going to do anything. Okay, so you're going to crank it up a little bit. You don't want to heat it up too much. If you heat it up too much, then the primers will fall off. Okay, so actually there are math involved in figuring out the exact temperatures, but basically it's like very hot, a little bit cooler, and then a little bit warmer. That's one cycle. So at the end of one cycle, you will have 4 copies. And then now you will do it again. Then each of the 4 copies will separate, and then the primers will bind to the 4 copies, and then the tag will copy all of them, and then you have 8 copies, and so on and so forth. And before you know it, you would have cloned the DNA that you want. So if we're trying to make insulin, we'll be cloning the insulin gene. That's PCR in a nutshell. Any questions? All right, so we are done with the first 2 steps. I'm just gonna tell you about some random stuff, and then we're done. We'll be done. First of all, if you have 10 copies of DNA and you do 4 cycles, how many copies will you have afterwards? What do you guys think, huh? Okay, that's good try. Anyone else? So start with 10. It will double every cycle. So times 2, that's the first cycle, second cycle, third cycle, fourth cycle. 2, 2, 2, 2 gives you 16. And then times 10, that's going to be 160. Oops, 160. So basically it's starting number multiplied by 2 to the n, where n is number of cycles. $160 might sound like a lot, but we need millions of copies in order to feed them to the bacteria. Okay, let's talk about the PCR for COVID test, which is a special kind of PCR called RTQ PCR. So um, if you're not sure whether you have COVID or not, well, nowadays you can test it at home, right? I don't think they hand out these kits anymore at the pharmacy, but it used to be free and then they hand them out like candies. Remember this one, right? That looks like this. And then you would add your sample here, and then you would wait to see whether you have 1 line or 2 lines, right? I assume you guys have done this before. I've done this a couple of times myself. This is an antigen test. Antigen test tests for viral proteins. Okay, do you have the COVID proteins in you? Okay. So it's easy to do, but sometimes it doesn't have the same sensitivity as a PCR test. PCR test, on the other hand, instead of testing for viral protein, you're testing for viral genetic material. Okay, so this takes some time. You have to send it to the lab. You cannot do it at home. You don't have the, ingredient or the machine to do it. And so Another thing to understand is we start with DNA, which you change to RNA, and then translation will allow you to build a protein. That's how information flows in a cell. However, when it comes to viruses, there are two kinds of viruses. We have RNA virus, which is what the COVID virus is. The COVID virus is actually called SARS-CoV-2. And then there are like DNA viruses. Okay, DNA viruses are rarely of concern because they don't really mutate, they don't really change. Chickenpox is a DNA virus. Okay, so you get chickenpox once, you're pretty much protected for life. Okay, or if you get the chickenpox vaccine, then you're not going to catch chickenpox. Right? Those are DNA viruses. They don't really change. Right? Polio, for example. Right? You get vaccinated, you're good for life. But for RNA viruses, they don't have a spell checking mechanism and they make mistakes every time they copy their DNA and they evolve rapidly, which is why every now and then you hear there's like a new COVID variant of concern. That's basically telling you that the virus has mutated and it's different from before. And because it's different from before, the vaccine you got previously might not be as effective against the new one, right, as the previous one. So that's why you also need to get new shots every year, right? Same thing for influenza, right? Flu shots every year, right? So the RNA virus will infect the host, and it actually needs to change its DNA back, change its RNA back into DNA first. And the process is called reverse transcription. Okay? It's opposite of transcription. Then the DNA will be transcribed and translated using the ribosomes and everything else within the host. All right. So what this does is when you do the swap, if you have the virus, you will swap up the viral particles along with it, and you would add it to the solution, and they basically would dissolve the virus shell, but leave the genetic material alone, and they would supply the RT. RT here stands for reverse transcriptase, okay, which is the enzyme that will allow for reverse transcription. Right? So it causes the viral RNA to change back to the DNA. Now, again, this is only going to happen if your sample actually has the viral RNA. Okay. So assuming it does, it will get changed in the DNA, and then you will do PCR the way I've told you. The primers that are designed for testing COVID would recognize the viral genetic material only. Okay, so if there's no virus in your sample, the primers will have nothing to bind to. It's not going to recognize your DNA, and then the test will become negative. Okay, which is why it's very powerful. And one of the first thing that scientists needs to do when there's a new virus out there or something like that is they are in a race to sequence the virus. Once you sequence the virus, then it gives you a way to detect them, right? Using things like PCR. Anyways, the Q here stands for quantitative PCR. Right? It allows you to track the number of copies as you're cloning them. So here it's a graph that is being plotted as you run the samples, okay? Each line here represents a different sample. And just to keep it simple here, I know these numbers doesn't really quite make sense to you unless you learn, logarithmic scale before. Anyway, long story short, just pretend this is like, 10,000 copies. Okay, that's basically what the y-axis means. So how long does it take for you to reach 10,000 copies? It's what you are interested in. So that's a threshold. Well, let's take a look at this sample. Right? This sample doesn't take very long before it reach 10,000 copies. So what does it mean? It means this actually has high viral load. Right? There are a lot of viruses in this sample, a lot of viral DNA fragments to begin with. That's why it didn't take that long to reach 10,000. Whereas, like a sample over here, it takes 25 cycles of PCR to reach 10,000 copies. So you didn't have, a lot of samples to begin with. You need to copy over and over and over and over again before you get 10,000. So this last sample right here probably doesn't have a lot of virus in it. This person is probably recovering already. Okay. So by doing qPCR, not only does it allow you to confirm whether a person has COVID or not, it also gives you information as to like where are they at in terms of the infection. Is this like really early on, right? If so, you probably should like isolate that person, or like is this later on? If it's later on, it could be more worrisome because that if that person didn't know and they have been walking around, spreading to other people, then it's actually more concerning, right? Certainly more so at the beginning of the pandemic, right? Nowadays, we are more used to it, and most people are protected because of the vaccine. So that's how the PCR works in terms of testing with the COVID stuff. And that's all for this class. Anything else you guys want to ask before we say goodbye? All right then. I will see you guys on Thursday. Remember, the quiz has been moved to next Thursday, so it give you a little bit more time to study. Have a good day, guys. Take care. Bye.